Good morning and good afternoon, everybody. You've just come to Cyberhuman Frontiers, and it's December 19th, 2020. We have a continuing voyage into the spooky world of quantum physics, and the more research is done, the more compelling that it becomes to us, and it will be to you as we show you what's being found, what connections are being made, what relationships between the world of cosmological physics, that's outer space, and human biology. And it really is a brand new frontier. There are two ways of thinking about things. As it comes to mind, as I thought about this before the show, that we can think of things in either concrete, three-dimensional physical terms, or we can think in more abstract terms, which sometimes require mathematics to express them. That's a little bit beyond what I'm good at. I'm not a fantastic mathematician, but I can follow those people who are very gifted with mathematics, especially if they come up with graphical representations of what it is they're talking about. What do I mean by all this? It is bringing the world of the invisible into a visible picture that we can wrap our brains around. And when we're talking quantum physics, that's kind of what it is. I think that's almost enough of an introduction as to why. So we do need to appreciate those people who are so well-versed and gifted with mathematics, because they help bring that into a realm of understanding for ordinary people like me. And there's an extraordinary person, a little bit like me and a little bit unlike me, who's here too, and that would be TMC. How are you tonight, TMC?
I'm great, Indy. Thanks for asking.
Good. I'm glad to hear that. Every day is a new adventure, especially these days. Every day is really a new adventure. Wow, man. All right. So before we get started, we do know that our streaming platforms, particularly YouTube, are changing some of their policies for community guidelines and such. So I'm just going to bear with me for just a second. I'll try to read it as fast as possible. But due to the Copyright Act of 1976, allowance is made for fair use for purposes of comment, criticism, news reporting, teaching, scholarship, and research. The community guidelines disclaimer is this. The points of view and purpose of this video is not to bully, harass, or incite fear or violence in any way, but rather to share opinions and thoughts with other like-minded individuals curious about the subjects, to encourage conversation and awareness. The opinions expressed on this stream belong solely to the persons espousing them and are not an expression of the channel Cyberhuman Frontiers. This video does not claim to give medical, legal, political, or financial advice. Notwithstanding the provisions of Sections 106 and 106A, the fair use of copyrighted work, including such use as reproduction in copies or phonorecords by any other means other than specified by that section, for purposes such as criticism, comment, news reporting, teaching, scholarship, or research, is not an infringement of copyright. This video and our YouTube channel in general may contain certain copyrighted works that were not specifically authorized to be used by the copyright holder, but in which we believe in good faith are protected by federal law and the fair use doctrine for one or more of the reasons noted above. Thank you for letting me read that. And now let's get right into it. We've been exploring the frontiers of quantum physics. And now we're going to do a little bit more tonight, focusing a little bit more on human DNA. Multiple research institutions and scholars all over the place have observed and begun to quantify and mathematically express these phenomena of DNA. So, for example, DNA exhibits quantum filtering characteristics, and we'll get into what that means a little bit more. With light photons. They've observed it specifically in its effects on light photons. It is a fractal antenna. And at very low levels, DNA, through its molecular reactions that go on, it emits and receives light. And it may function in some manner as a quantum gateway. And we have a lot of DNA in us. So as we become more articulate in the lingo and more familiar with the general terms that are used with artificial intelligence and quantum physics, as you follow us through our episodes, you're going to start to find that it's almost obvious that our body's cells, particularly those in our brains, have more capacity than we can actually graphically chart out in the tangible three-dimensional world, and that it's probably far beyond what we normally would refer to as mere thought processing. It is our opinion, and once again, it's our opinion, but we'll show you what substantiates that a part of us, that's each and every one of us, all of us, quite possibly has an essential part of our intelligence, and some aspect of us may exist in an extended reality. And that can explain a whole lot of things, such as intuition, other types, what happens to a person when they die, where do they go, sense, like extra sensory perception instances that people have indicated from one time or another. There are many, many anecdotal accounts of that people have experienced things. The linkage that seems to occur between identical twins, somehow they seem to be linked in ways that are just not obvious to us. But there's been a lot of evidence that just occurs over and over again. So that's why we say that, and with mounting experimental data and reaching the limits of what we can see in that tangible world, more and more scientists are looking in this direction to start to map out the unknown, the unseeable. It's invisible. So that being said, I think we'll get into something. Where do you want to go with it initially? I think maybe what we might do is play a little bit of that first video that I was thinking of, TMC, or do you want to do a little reading? You want to do that first, right?
Yeah, we could do that. Let's set the table with
okay.
construct.
Yep. Well, there is what seems to me to be an extremely intelligent lady named Ulrike Granoger, and pardon me if I didn't quite pronounce her name correctly. It seems to be some sort of a Scandinavian or a German, probably a Scandinavian name. And she, in very, very few words and very succinctly and elegantly expresses them. And so what we have here is a little video that she did, and she is getting into exactly what we just talked about. And it goes further than this, but she gets into something called the mathematics of the genetic code. So you see, this is her YouTube channel. We want to thank her in advance for letting us replay some of her material. And this is only a 5-minute video, but I think that if you take it step by step, this is where we start to get into that territory of the mathematical and the beyond 3D, the beyond three-dimensional, the beyond the tangible. And she's going to, well, let's play it and see what you think. Here we go. Right now.
These three properties describe a type of a chemical space. That's the term, not the use of the word space. And the set of 20 amino acids covers this space perfectly. They found that among 1 billion...
What she's already doing is she's starting to show us that there is a kind of a geometrical precision among the amino acids which make up all the proteins in our body. And what she's observed is that there is, once again, an elegant type geometry about this. So here she goes.
In random sets of 20 amino acids, there is only 6 that have similar or better coverage of the chemical space properties. They also found that other sets of 20 amino acids or even of less amino acids that contain at least one or two of our current coding amino acids would be much better in adaptivity than sets, random sets that contain none of our 20 amino acids.
So right here, what we see is these letters, A, C, T, and G, adenine, cytosine, thymine, and guanine, which compose a significant part of the backbone of DNA. And what she's doing is she's laying the groundwork for the idea that there is a chemical space that a unique small group of amino acids occupy that lend them to being perfect, the perfect molecular structures to be what she continues to talk about in the next little piece of this.
And what is more, none of the artificial sets would use less energy for the same capacity as our natural 20 amino acids.
Okay, now that's significant. Now she's beginning to lay the groundwork for there being a kind of an efficiency about nature, and that the particular molecules that are, they are just inherently part of our natural structure at many different levels. These amino acids are the ideal ones because they most efficiently work together in this chemical space that she's talking about. It starts to suggest that it's not accidental and that we've had many efforts at creating artificial amino acid analogs, and they don't work as well as the ones that nature built itself. And that's probably no accident. She goes on.
This means that life on Earth has somehow chosen the best possibility among billions of different sets, and it may.
She gets into what the billions of possibilities would have been for the possible configurations of atoms and molecules. And the number of possibilities that there are is literally mind-boggling. You'll see the number 10 to the exponent 48. 10 to the 48th power is an absolutely gigantic number of the possibilities of different configurations of all of these atoms that could be in an amino acid, which we're seeing out around the edges here, or in a larger protein, a biomolecule. Now, just to put 10 to the 48th exponential power into perspective, there are only about 10 to the 16th grains of sand on the entire Earth. So 10 to the 48th possibilities is, I can't even comprehend how big that number is, but the chances that this all got put together by random chance are extremely small, because the chances of having a wrong combination or less than the best combination would have been extremely huge. She goes on.
I mean that life beyond Earth may look very similar or use a very similar set, a type of an amino acid language that is universal. One of the...
And I want to just stop on that for a second. There is a universal language maybe a couple of them in the entire universe. This is not all Earth-centric. There is a chemical language in the universe, and that is the elements, the fundamental elements of the periodic table, including hydrogen, carbon, oxygen, nitrogen, and a whole bunch of others. They are all common elements in the universe. Okay, and there are certain mathematical principles and physical laws that are an outgrowth or can be expressed by those mathematical principles that are common throughout the whole universe. What she's saying here is that these amino acids, being a higher function of the mathematical laws and the physical laws, are also going to be common through the universe, the same way that the fundamental elements that make them up are, which would suggest that if we ever do run into ETs, they're going to have a whole lot of common chemistry with us. It's kind of where she's going.
Co-authors of the report saying, quote, life may not be just a set of accidental events. Rather, there may be some universal laws governing the evolution of life. In a fourth symmetry, Shabak discovered the number 37 as well as rotation in the following way. In the same way of counting the nucleons.
Now, I'm going to stop it right here because this is where it gets kind of technical, but fortunately, this is a short video. So if everybody can just manage to just listen to it and maybe hope that some of it sinks in, maybe you'll understand it better than I do, but some of the numerics that she gets into a little bit beyond me, but it's only a couple minutes of it. So here she goes. The thing to key in on is the commonality, the common factors and the patterns. That's the big takeaway from what she's going to cover here.
In standard blocks and side chains of amino acids, he's now looking at those amino acids that...
Sorry to keep interrupting, but these codons refer to parts of a DNA molecule. There are two types, the larger type, which are adenine and guanine, and the smaller type, which are thymine and cytosine. And this is what makes up our DNA. So she's talking about DNA. It doesn't say codon, doesn't say DNA here, but that's what this is. This is DNA.
The codons they're known sequences. So they're combinations of base pairs that are like base combinations. They're root combinations of base pairs of DNA.
So you're right, eh? That's what she means right here when she's got these groups.
That's how they're charted out, right?
She's got TTA and TT. Yep.
Right. So these are base groupings of nucleotides, which are the building blocks of DNA. You see that in the top line and on the left-hand side, TCG. Right. And you also have U, but that's really when you're working with RNA. So we'll just stay with what's on this chart here. So you've got the base compounds, the base nucleotides, TCA and G, but then you have specific combinations of those. And she's tabled it out as double base codons, which is unique combinations of the A, C, T, and G in their root form that can be combined in different combinations to make up proteins and
amino acids. And she's listing amino acids here. Here's phenylalanine. Yeah. Here's these are amino acids. Here's leucine. Here is valine. Here is serine. Here is proline. Here is threonine. Here is alanine, lysine, aspartate, glycine, and arginine. These are so in other words, these triple base pair codons. I'm glad you mentioned this because this is the direct building block of those amino acids, which is what she's talking about. Yes, sir. That was important. So check out what she sees for emerging patterns here.
He's now looking at those amino acids that share two identical bases or letters. The total number of nucleons in the radicals of AA amino acids plus the total number of nucleons in the radicals of G amino acids amounts to 999.
She's beginning to trace out the pattern for us. So now you see the names that those triple codons code out for those amino acids. And now she's starting to show us this numerical, amazing stuff.
And again, the total number of nucleons in the radicals of T amino acids, as well as the total number of nucleons in the radicals of C amino acids, amounts to 999. The total sums of nucleons in the standard blocks of each of those four groups amounts each to a multiple of 37. 592, 666, 666, 666.
So what this is beginning to suggest is that there is something of a higher order logic to what the foundational components, the adenine, guanine, cytosine, and thymine, reflect in the construction of amino acids. So think efficiency, think pattern. She goes on.
And of course, their total sums again are divisible by 37. What is more, if you look at the positioning of each of these groups in the table of the 64 triplets, you realize that the double A and double G triplets and the double T and double C triplets are in an exact rotational symmetry to each other in the table.
Somebody maybe have to help me out with this, because she lost me on that. I don't know what she means by a rotational symmetry. We're just regular people like you guys. Well,
you've got the symmetry in that the fatter, shorter box, right? Those are opposite each other, and then you've got their corresponding thinner, longer rectangular box in the opposite. And so when she calls it rotational, it's the fact that they're not stacked on each side, that the yellows aren't on one side and the purples aren't on the other. You have a symmetry in that you have a yellow and a purple, and then the inverse of the shape orientations of yellow and the purple on the opposite side. So the symmetry being, if you were to draw a diagonal line between the two, you get the same picture on both sides, a mirror.
Complementarity. They're different, but there's a symmetry in their complementarity, maybe, kind of, sort of.
And you said that those were the larger.
The purines are the larger ones. The pyrimidines are the smaller ones.
Got it. Okay.
Yeah, I think you could just, with the chart, you could just draw a line. Okay, all right, okay, I got it. Thank you.
To each other in the table. And finally, if you look at the sums of the nucleons in the radical of each of these double base groups, 535, 464, 547, 452, you arrive at the most amazing symmetry in the summation of these digits, both a symmetry as well as a cyclical permutation. Each of the sums, whether read from the left to the right or counted from the right to the left, and in each of the permutations of each of the numbers, gives you a triple of 666. By itself, it is a feat to find 4 different, seemingly unrelated numbers that in each of their permutations and sums gives you a triple of 666. But to have this number from the nucleons of atoms.
So just to keep in focus, this is derived from summing up the atomic weight of each atom and coming up with the molecular weight, and then taking that molecular weight, the molecular weight number, and putting it on, where'd that chart go? Putting them on this chart. And the likelihood of these things being equivalent would be mind-bogglingly unlikely, just by accident, is what she's saying.
That in each of their permutations and sums gives you a triple of 666. But to have this number from the nucleons of atoms in molecules that make up the genetic code is mind-blowing.
I read this, and it does get into a mathematical expression of it, and people who are really good at that could probably read this and make more sense of it than I could. But I've even got it up here, I think, someplace, but we don't need to get into it right now. So where we're going with this, can you tell us where we're going with this? There's something about a kind of order. There's symmetry. TMC, expound if you're so inspired.
No, I'm kind of stuck on the whole 666 thing. I mean, I'm sure that's shocked quite a bit of people watching this so far. But I've heard that analysis before, that the number of the human is 666, and you can misform or misquote that all day long out of the Bible, that being the mark of the beast or whatever. But
Right.
they actually they give the number, they give a, how do I say this? They give a name to the man, and the number of the name of man is 666. And I think that's actually stated in the Bible. Now, the question then becomes, when was that put in? Why? Where did the information come from? Did they know about DNA? If they did they understand how it broke down into these universal mathematical additions where all the permutations, regardless of what the permutations are, if you add them up, you end up getting 666. It's pretty remarkable. And it my mind was going to.
I went there too.
Consideration, right? Where did that information even come from in the first place? Because if it was provided to us, by who then is the question I have. And if we figured it out and this stuff is dated, this information is dated to be recorded, thousands of years ago, why are we now just sort of coming up with this conclusion in 1998 that these mathematical similarities or coincidences are actually discovered in 1998? So I'm kind of torn.
Given that how this number and the sum of it, 1998, is arrived at by concrete, incontrovertible molecular weights of amino acids, you can't argue with the idea that these add up, that the right combinations of these add up to that. And they do it repeatedly. So that's just not debatable. It is what it is. And so I went, what's that?
You go back to that paper. Is there a date on that paper? That 1998 are they date stamping when these?
Oh, I've got it loaded here someplace.
It says 2010 on the paper.
Yeah, I've got it in the tabs up here somewhere. Let me see where it is.
Because it was in the late 80s or 90s is when they actually went through the whole human genome. And that's why the 1998 stood out as a date to me more than anything else.
Well, what we got, this isn't I can get that exact one because I did have it up.
We don't need to.
But kind of another way of looking at this thing, she did a wonderful job. My compliments to that lady. Life uses 20 coded amino acids, and now we know what CAA means, right? Coded amino acids to construct proteins. This set was likely evolutionary standardized from smaller sets as organisms discovered how to make and encode them. And once again, keep in mind efficiency. Little critters that are going to become more complex animals are only going to do it if it works efficiently. If it doesn't, they won't survive. So scientists modeled how the adaptive properties of the coded amino acids evolved over time. And they found that sets containing even only a few coded amino acids were better than an enormous choice of alternatives, suggesting each time that a modern coded amino acid was discovered, it bootstrapped the set to include still more coded amino acids. So now this expands based on what, oh my God, I forgot her name. What was it? It was Grudenberger. Oh, shoot. What the heck was her name? Ulrich Grunauger. Sorry, Ulrich. She went on to say this probably expands to any potential life that you might find when you get in your spaceship and drive to another galaxy. You're going to find it's more than highly likely that other life form that you run into, that ET, is going to be comprised of proteins that are made from the same group of amino acids because it's what works. And that group of amino acids is an outgrowth of laws of physics, which is why those amino acids are the way they are. So this goes, all life from bacteria to blue whales to human beings uses an almost universal set of 20 coded amino acids to construct their proteins. This set was likely canonicalized. Is that a word? Canonicalized or standardized during early evolution. Before this, smaller amino acid sets were gradually expanded as the organism grew to develop new synthetic proofreading and coding abilities. And so we're at that level now where we're doing deciphering of the proofreading, and we're now working with doing synthetic coding. And I think that what we're going to find is that when we try to synthetically code something that's not efficient, that is not most likely almost an exact duplicate of the natural amino acid structures and shapes, it probably isn't going to work real well. It goes on. There are millions of possible types of amino acids that could be found on Earth or elsewhere in the universe, each with its own distinct chemical properties. Scientists have found these unique chemical properties are what gives biological proteins, the large molecules that do most of life's work, their own unique capabilities. And now I'm going to go on to this paragraph, which is the real amazing part right here. The team set out to ask the question, smaller coded sets might have been like in terms of their chemical properties. There are many possible subsets or other presently uncoded amino acids that could have comprised earlier sets. The team calculated possible ways of making the set of 3 to 20 amino acids using a special library of 1,900 structurally diverse virtual amino acids. They computed and found that there were 10 to the 48th ways of configuring the sets of 20 amino acids. 10 to the 48th is a number I can't even think about. It's way big. In contrast, there are only 10 to the 19th grains of sand on Earth and only 10 to the 24th stars in the entire universe. So the point is, there are so many possible ways that things could have been combined. And looking at it from a computational approach, you can come up with really big exponents. And so they go on to their summary. You want to read a little of this, or should we skip it?
It's up to you, man.
Well, what we've discovered is, in a few words in summary, is that the structure and nature and configuration of life is no accident. Now, we haven't proven it directly, but when you've got 10 to the 48th ways of doing it wrong and only a few ways of doing it right, and it happened to have come out right, and it's also something that's going to be in common with life that you're probably going to find anywhere, from a blue whale to a bacterium to probably any other life form that you might run into anywhere else. That is pretty profound. And that's kind of where I wanted to get to with that. So this is an intro to whatever you'd like to talk about next. Just okay. Life is amazing.
Well, we've been trying to get through this document. And this document talks about quantum mechanics and biophotons, right? So those of you that have watched, you'll kind of be familiar with this. We've danced around this document. We've gone through it quite a bit. And we're at the point where we're going to start looking at DNA and what it really can do. Some of the studies they put together it kind of explain some of the cool things that DNA does. And it's its own unique little computer processor. I think I pulled the wrong doc. This is only a single page. Why? Hang on a sec. Let me...
There we go.
Yeah, that's better. Okay. So we're at DNA. And we're gonna read this because it's gonna set the table for upcoming episodes. That get into some of the other cool stuff DNA can do. And from what we've seen come out recently, DNA can basically, you can take a holographic image of DNA, transmit it over the internet, and put it into something else and change the DNA just by transmitting a holographic image of DNA.
So do I understand you correctly that a holographic image is something that can be converted into a string of digital data, a long string, probably a big string, but
it's pulses of light. Yes.
Once you get those pulses at the other end, you can reconstruct that whole holographic image.
Right. That's what hopefully we'll be able to show once we finish parsing through that video, and we could do it in some sequence that doesn't take up, the whole 2 hours and doesn't have a whole bunch of the technical stuff, just more of some of the provable experimentation that was done. And the implications are huge. Outside of DNA being a great communication antenna / storage vessel,
information storage vessel, really high capacity information storage vessel.
Information processor, yeah. It can also be used to, replace things like vaccines and age regress or reverse age other types of diseases and whatnot. And so the therapeutic implications are huge. And all of this really centers around the quantum mechanical effects that are exhibited when you start studying and understanding DNA. So that's why we're going to go through this document as kind of just an overview of what some of the possibilities are. So here we go. A quantum dot, right? DNA. It's a single atom-like light emitter, which can be used in quantum light sources. The property of a quantum dot can vary dependent upon temperature and external electromagnetic fields. Quantum dot is a nanostructure containing highly tunable properties that may have applications in quantum computers. Its unique photophysical properties may allow for optical encoding. Spectral tuning can be modified by curvature of 1 micron diameter microfiber when coupled to a single quantum dot. So this is lab experiments to show what can be done with something at this scale, nano-sized materials. So placing the correct or the right resonant frequency quantum dot at the right place is required for on-demand reconfigurable photonic crystal resonators. The RPCR is applicable to a quantum dot single photon source. A photonic crystal is a nanostructure that affects the motion of photons. A resonator is used to generate electromagnetic waves of specific frequencies. By employing the contact of curved microfiber, 1 micron in diameter, is possible to achieve spatial relocation and spectral tuning of the resonator, as well as a photon collection efficiency. So you're like, why all that? DNA programmable methods are used to prepare 3D photonic crystals as precursors of assembling nanostructures into very complex structures. So they're saying that they use DNA as the scaffolding or as the subsidiary lattice or the building blocks to put these nano photonic crystal structures into the right place to start being able to create complex geometries to bend and manipulate light, to manipulate photons. And this is done in the quantum information science. It'd be a good general term to search. If you're curious about quantum information, look up quantum information science, and that'll kind of put you on the right path. So it is not known if the quantum dot can encode by directly tuning, nor is it known if these discoveries in the discipline of physics will be applicable to biology. It is possible to exploit quantum non-locality to transmit usable signals that can travel faster than light. Quantum entanglement can be injected into optical fibers and mysteriously interact at a distance. This is kind of an important sentence because what they're getting at here is the ability to create quantum entangled photons over a fiber optic network and be able to have instantaneous communication faster than the speed of light if you can maintain quantum photonic entanglement through those optical fibers. And now we're starting to look at what maybe a quantum internet mechanically would look like.
Right.
Quantum teleportation is the most surprising application.
Whoa.
Yeah. Communication looks like episcopal, bystander, or biophoton between cultures or animals can be viewed as quantum teleportation, and DNA can be the command and control center for the cells.
And how does that work? It's got to be because there is entanglement of some sort. There's some relatedness.
Yeah. And so here's where DNA kind of gets pretty cool. We talked about these crystals and quantum dots and complex lattice structures using DNA as scaffolding. Well, DNA itself is a liquid crystal. It's its own thing. It's a liquid crystal lattice type structure that may store and emit biophotons. Hang on one sec. I'm going to scroll this up and just keep reading. I got to mute for a second.
Yeah, that's good. Yep. I think you're muted right now. You might have done it by accident. So yeah, while he's getting that thing, getting it going the way he wants it to, yeah, big hellos out to everybody. And it's, it is, oh, you are back.
Yeah,
it is truly profound that when just good researchers in a biolab and some physicists get together and they keep looking at things and they keep analyzing and they get deeper and deeper and they start to find some of these emergent organizing properties about them. And we have possibilities going forward. We're living in such an amazing, brave new world going forward that we just feel compelled to bring this to you because it can really change the way that we look at life. It can really help us to be better informed about our place in the world, our place in creation. I don't know if you're a religious or spiritual person, but it helps to give a person a lot of good stuff to think about. And knowledge is very empowering. Anyway, I could go on forever. Are you back where you want to be now?
Yeah, I had a emergency real quick, so I had to take care of it.
Okay.
We'll want to just continue reading. I mean, we're getting to this part where DNA is basically, they're going to talk about it being a computer chip in its own way, a light processing computer chip. And so anyway, so we'll keep going. So about 95% of human DNA is not involved in protein building. It's not necessarily junk DNA, but it's active within a quantum state to transfer information. So we're talking about DNA being a central command and control like computer that's able to interact with all the cells in your body at the same time through quantum teleportation, right? So it says here that signals can be read out by DNA without any essential loss of information. DNA is a complicated electronic biological chip that communicates with each cell and with other cells of the body. The DNA code is used for hypercommunication, which is a data exchange at the DNA level using genetic code. They talk about the Watson-Crick chart graph rules.
Yeah, it's a higher level than just the simple day-to-day business of, making proteins, making energy and burning calories. It's a higher level than that.
Yeah. So it says here that the Watson-Crick, which is the standard coding for DNA, that basically lines out the different combinations and the different rules and structures as it relates to the combinations of A, T, G, and C, these nucleotides that make up the base pairs for DNA.
Read that next sentence. Look at that. That next sentence. That's whoa.
Yeah, it's saying that Watson-Crick is only correct about 5% of the time as it relates to the nuances of DNA. And so it says laser light is generated in DNA. Right here. Laser light generated in DNA is a key element in this information transmission system. So notice above we were looking at quantum dots and quantum entangled photons over a fiber optic network, and then setting up a fiber optic piece of fiber optic cabling, so to speak, to be able to interact with quantum dots to function at this level. And what they're getting at here is that DNA does it naturally. DNA generates its own laser light, and it's laser light because it's coherent light, and it's because DNA is spiraled, and it becomes what they call exhibits chiral-like properties. So when the light is generated, it's there, and it only moves in one particular polarized way, one particular way that it spins.
It filters
or rotates around. That's a better word. It's a light filter. It's like a polarizing filter naturally. And because of that reason, they're able to say that it's laser light. It's coherent light.
Not only is it a filter, but it also generates light.
Yeah.
So when we think of one of those now commonplace laser pointers that people use, did you ever stop to think that at a very tiny scale, every little piece of DNA in your body is a tiny little laser pointer, and it's receiving and transmitting coherent light for some purpose. We don't know a whole lot about what all these purposes are, but they're way more than what we would have normally just thought of as all of the normal metabolic processes of an organism. It's way more, and we don't know what it all is yet, but it's huge. It's huge.
So those codons that we were looking at earlier, those base primary combinations of AT, C, and G, those codons, the three minimum nucleotides put together, it says here that they may actually form words and sentences, just like our ordinary human language follows grammar rules.
Hey, can I stop you for a second?
Yeah.
Point out there in the chat, and I'll quote it. I've previously reviewed this info, and it makes a slow-moving, agonizing admission of intelligent design, also known as a creator. And it's kind of like that. It's like the more that the scientists and the physicists work toward this, it's like they paint themselves into the corner of having to almost acknowledge that there's got to be some higher organizational intelligence. And, I would use the word creator. There is a prime mover, a creator, some force by some means of, you can use the word creator or God, that put all of this, set all this in motion in a way that is so elegantly organized. And it's not just us. It's bacteria. It's the universe. There are common laws of chemistry and physics throughout all of creation, for lack of any other word. Common laws. And quantum physics is involved. Quantum phenomena is part of it. So this is, anyway, there I go.
Yeah. So since the DNA is found to have specific grammar syntax and semantics akin to our human language, it's indicated that our currently restricted understanding of DNA, serving only for the coding for reproduction of proteins, for the chemical makeup of organisms, is only part of the story. And here we get into some of the other cool stuff. Ultra-weak biophotons, light, may represent a complex cell-to-cell communication system that relies upon speed of light or greater transmission, that teleportation or quantum entanglement, based on data in DNA. The physics of light.
Hold it right there. Let's just clarify for everybody for a second. You can have a photon and it will travel at the speed of light. But if you have one particle that gives rise to two photons, then in most scenarios, those are entangled photons and they are related, and they now can transcend the normal speed limit of the speed of light, and they can also transcend the time to traverse a distance as well. So
I'm going to kind of
disagree with me on that?
Part of that, yeah. I don't think that they can necessarily transcend the speed of light as it relates to their travel time per se. But when they're separated farther than
You're right.
the speed of light, they can communicate with each other faster than the speed of light.
Right.
The interaction between the entangled photons is instantaneous for all intents and purposes.
You're right.
Their travel time doesn't necessarily mean that they can travel faster than the speed of light. They've got subparticles that they say travel faster than the speed of light, I think, to try to define some material explanation for this instantaneous communication. You get into quarks and condensates and all that other stuff, and we're not necessarily going there. Neutrinos comes to mind. I think that lady talks about neutrinos in some of her other discussions, as a matter of fact. But for all intents and purposes, it's the instantaneous communication that exists when photons or electrons become entangled.
Yep.
They can travel to different places and still interact with each other instantaneously as if they were right next to each other.
Yep, that is what it is. Yep.
So it says here that light is the most efficient and the fastest mediator of information in the world. The coherent property of biophotons has a profound effect on their ability to influence information transfer. Frequency coding. Frequencies of light. So red light, green light, blue light, yellow light, infrared light, purple light, ultraviolet light, different frequencies of light, right? Frequency coding gives light a capability of encoding information from DNA and biophotons. The optical resonator is required to store light within a very small confined space. The ability to trap photons and influence the propagation of light plays a significant role in quantum optics. Photon signaling among cells and animals indicates that cells can talk to each other. Biophotonic signaling is used in the reception, transmission, and processing of electromagnetic data, perhaps with some of the same transmission features of fiber optics.
So they now introduce the term electromagnetic data, and photons are a form of, if you look at them in terms of waves, they can be expressed, and it's valid to express them as electromagnetic properties as well.
Yeah, definitely. That's photons would primarily be, it's a root form of measurement on the electromagnetic scale. So radio waves are photons, millimeter waves are photons, microwaves are photons. Light. So
smallest indivisible particle of a wave becomes finally a photon, right? You can only divide a wave up so many times before it's got to be a discrete size piece, and that is the size value of a photon.
Yes and no, because you could break photons down even further than that. When you start, smashing stuff together, you can get, like I said earlier, neutrinos and gluons and all that other stuff. And so there are smaller elements of matter, but they're not necessarily measurable for any length of time to be considered matter. Or stable.
Would they I guess matter. In some sense they would still be part of the electromagnetic wave, but if you want to divide an electromagnetic wave, a sine wave or any other shape, into smaller and smaller segments, at some point it becomes steps of photons. There's I think I remember that from something that we read.
Yeah, the root measurement of electromagnetic energy is a photon, and then a photon has an energy rating based on its vibration. So the shorter the wavelength, the higher the vibration, the more energy per photon something has. So X-rays, gamma rays, much faster wave. And what I mean by that is the oscillations up, down, up, down, up, down per second they're much more of those when you compare an X-ray to a microwave, or when you compare an X-ray to a radio wave. So radio wave, one photon of something in the radio wave frequency band has much, much less energy than one photon emitted as a gamma wave or an X-ray wave.
Yep. Higher frequency, shorter wavelength.
More energy per photon.
Hey, you want me to read a paragraph?
Yeah, go for it, man.
All right. So this individual, Pop, spent nearly 20 years studying biophoton emission. He found that DNA is the main supplier of biophotons. He discovered that DNA is a harmonic oscillator, an oscillating system with its own particular resonating frequency. Biophotons represent a wide variety of frequencies which seem to originate from DNA and may be concentrated in the DNA of the cell nucleus. According to Pop, light can be stored in tissues and gradually released over minutes or hours. Well, that's fascinating in itself. Pop concluded that biophotons appear to communicate instantaneously with all the cells of the body in a synchronous wave of informational energy. If you gather what we're reading here, this is just a whole different way of thinking about how our bodies work or how any organism's body works. Pop put forward the hypothesis that biophotons analogous to a laser are emitted from a coherent electrodynamic field within the living system. He believed that biophotons may represent a wide variety of frequencies which seem to originate from DNA and be concentrated in DNA of the cell nucleus. Accordingly, light can be stored in DNA and released over time. It must be stored as some form of energy, and it must have encoded in the DNA exactly what nature of photon or what frequency of photon and what coherent orientation it's going to be released in. That was me saying that. He concluded that biophotons appear to communicate with all the cells of the body instantaneously in a synchronous wave of informational energy. Overall, there is a relatively large amount of literature about DNA and its ability to create photons in a coherent state. Light can, as a bunch of photons, be manipulated in many different ways. Beam twisting using 8 beams of light causes the beam to exhibit DNA-like helical shape that is propagated in free space. Multi-party clusters of photon entanglement of up to 8 spatially separated photons has been demonstrated. With twisted beams of light, it's possible to send up to 2.6 terabits per second. So that's, a function of the speed of light and how fast you can transmit data if it's in the form of light photons. Light beam twisting can form a propagated helical shape that could possibly scan and encode parts of DNA and transmit enormous amounts of data. When DNA in test tubes is exposed to coherent laser light, the laser light spirals along the DNA helix as if it was guided by the structure of the DNA. Isn't that amazing? When you get your laser pointer and you point it at something, it goes in a straight line. And if it hits something that it's going to reflect off, it reflects off at the exact right optical angle. And when you consider taking laser light and shooting it into a test tube of DNA and it starts spiraling, well, DNA is photonically active, and it can emit light, and it can store light. I had a thought. I wonder if when someone does that in a laboratory, if the DNA is storing any portion of that light that it's illuminated with. I wonder.
I don't know. Storing, maybe it's excited or energized or manipulated some way. Not necessarily, when you say manipulate, it might not be harmful, but it's an interesting notion. I wonder if it understands the difference between a biophoton and an artificially created one.
And that would open a door to what might be happening with outside manipulation of DNA, in such sense as you've got your happy little organism just tootling right along, and it's got its DNA sending and receiving light. But then an outside source comes in, and, this is kind of where we get into the external sources of electromagnetic radiation, of which photonic energy is one, and that there might be ways of designing a signal that would influence the DNA or alter it somehow. And we start broaching into other areas, which we've talked about on other shows, such as the effect of electromagnetic radiation on our genetic material. And there's been a lot of study on that too. That's not exactly what we're talking about on this show, but it does touch on that. And I know that's kind of what you were thinking in the back of your mind.
Well, yeah, I mean, look at the possibilities of this. If they can twist beams of light in the right kind of way and create pulses of light that are many different light beams all twisting in a helical fashion to match up with DNA. Who's to say what could be done with respect to, turning on or turning off different pieces of the DNA or.
Well, let's go here. Let's go here. Back up to what Pop said earlier earlier in that paragraph, was that this is a means by which DNA is communicating from one part of the body or one part of a cell to another. So if you come in and you mimic something that would be like what DNA would normally be transmitting, but you do it by outside design by way of your computer equipment and your transducers, then you're going to change what that DNA might receive at the other end. It's certain that you'd be able to do that with the technology that they have now, some of which we've featured on these shows. They can do that. They may not have got it all mapped out for specific purposes and outcomes yet, but that that's right around the corner, right?
Yeah, especially when we start showing some of those experiments that they were doing.
And out in the chat, people are right on top of this. It's, electromagnetic pollution on genetic material. Yes. And some of it, a lot of what we live in, Celestial Dragon, great comments, is it's noise. There's so much chaotic electromagnetic radiation coming from, my goodness, you've got the radiation coming out of your router, you've got the radiation coming out of your phone, the electromagnetics coming from the cell tower, the radio antenna from the FM radio, the old FM style radio. You've got the higher frequency EM that's probably coming out of some of those new light bulbs that they make that are LED light bulbs, because those things do not run at just a steady state like a hot tungsten filament. They have a circuit in there that's driving them, and they're pulsing. And that circuit is a kind of a pulse width modulation power supply. So you got that. So all your LED light bulbs in your home, now that you've upgraded from the old ones, those all send out a little bit. Your computer, your CPU in your computer, the display on your computer, the flicker rate coming off the screen of your computer. We could go on and on and on. Your smart refrigerator, some of those things actually have an LCD display on the front of them now. All of this stuff. And then your Wi-Fi connected thermostat, your HVAC system in your home. I could go on and on and on and on. And then get in your car. There are multiple computers in your car operating. And those all have particular electromagnetic frequencies, which there's some leakage out of the wiring that's connecting them all, which you end up picking up with your body. So there's a lot of stuff. But now let's consider if there was a purposeful design of the pulses and the direction and the target of those frequencies to be aiming right at particular DNA, at particular frequencies that were maybe a little bit like what your cells and your DNA might normally want to do by itself, but they are just a little bit different. And so there's this thing that happens with waves whereby when there's a resonance, you start to entrain one object with another one. If you have two tuning forks and you start one of them vibrating, the other one will end up entraining to the same frequency after a short period of time if they're near each other. So that's called entrainment. So you can start to influence. And they were just hypothesizing. I mean, you heard our disclaimer at the beginning of the show. We're not saying anyone's actually doing any of this, but you certainly would be able to find ways to entrain the frequencies, the natural vibrational frequencies and photon output of human DNA with something external. That's just a certainty. For what purpose? I don't know. Could be for reprogramming that DNA, causing it to express a different way to mitigate a disease process, or it could be for other purposes, perhaps not such benevolent purposes. What do you think? inquiring minds want to know?
I think it's both. I think they have to go through the harmful to find out what works. I mean, you don't come up with a finished product without going through a whole lot of failure in anything.
True.
So,
trial and error, engineering, and engineering is often a very reductionist process. Initially, when they get something that they've actually got that works, it's really complicated. And then after a period of time, they start taking out all of the aspects of it that you didn't actually need. And after some period of research and development, engineering becomes what appears to be elegantly simple, but the complexity was in all of the logic in the way that it's put together. So things seem complicated now, but they're going to get better, faster, simpler, and more powerful as time goes on. And in some sense, one of the target objects of that is the human body, the human mind, our neurons, our brain waves, our EEG patterns. That's kind of a target of all of it. Hopefully all for good purposes, right? But maybe not. But it's good to know.
you want to keep going, or you want to move on to something else?
Could read some more. This is, to me, this is interesting. Little feedback from everybody out there. We'll go where you want to go. We kind of want to learn all of it and cover all of it sooner or later. Got some fun and amazing videos that we can show as well. I want to read a little bit more of this. Yeah. So this is like, quantum effects 101, let's just say. So when an atom absorbs a short burst of energy, it splits into two versions of itself, one excited and the other not. The following burst of coherent X-ray laser light scatters both versions, which then combine to form an X-ray hologram. Liquid crystal phases of the chromosome apparatus. Whoa, they're blowing me away here. Liquid crystal phases of the chromosome apparatus, the laser mirror analogs, can be considered as a fractal holographic environment to store localized photons. Oh my God, I'm scratching my head trying to wrap my mind around this. So as to create a coherent continuum of quantum, non-locally distributed, polarized wave genomic information. There is so much in those first three sentences of that. The fundamental notion is that the photon laser radio wave features of liquid crystals and DNA are stored for definite but varying times by means of laser mirrors as memory. Memory is an aspect of the genome's non-locality. What do you?
So do you remember the 2 meters of material that's wrapped up into 2 nanometers in a chromosome kind of a thing?
Yes.
Right. So that being a storage vessel and none of it necessarily overlapping, each having its own little compartment, being able to generate, emit, receive, or store light, and it being like the sections on a hard drive, where you have a certain amount of sections on a hard drive and you fill it up and your hard drive gets full, right? It's like that, but for DNA. And it says that it's done with liquid crystals that create holograms. And the cool thing about the holograms is that if you lose part of it, you need only a piece or one section that had the holographic information in it, and you can reconstruct the hologram. So there's something to be said for redundancy there with DNA, if in fact they are storing information as a hologram. If that hologram is distributed, once it's made, if it's distributed around, then that information can be retrieved from an undisturbed or a preserved section of DNA that hasn't necessarily lost that that information. Yeah.
Yep. Check this.
Yeah, good.
So what we have here is a non-overlapping pattern. So DNA has literally 2 meters long of DNA will be able to wrap itself up, and I'll get it here in 1 second.
Oh, yeah, it should show that piece. It's a beautiful piece.
Which piece? Which one you want?
The one that you're just talking about, where they talk about the DNA being 2 meters long, and then it's
coming right
up.
So here it is, right here, the human genome. And if you see this squiggly line thingy here, this is a believe it or not, 2 meters long of human DNA can be folded up in such a compact way that 2 meters of length will fit into, what was it, 2 square nanometers?
I think it was just 2 nanometers, yeah.
It's very small.
Space. Yeah.
So this is, and now we kind of go through it. They'll give us a little bit here.
Volume's down on the lower right corner.
Oh, I didn't know if we wanted to actually play the audio, but
why not? She explains it so eloquently.
Yeah, we could do that. Why not? All right, let's start it again. Let me share that audio. Here we go.
Phase 1. From there, move halfway to the corner of base. Every point inserts one of the nucleotides. What is found in these cells are long-range correlations extend across base positions, the existence of a superior context.
It's coming up in 1 second.
The correlations exist coding sequences of the genetic material, which is evidence that the 98% of seemingly useless DNA have organizing and context-generating information.
Okay,
An additional fractal or self-similar pattern of the DNA has been found in its 3-dimensional structure. Of so-called mechanobiology are slowly discovering that also the spatial organization of the chromosome is critical for regulating the genome. 3-dimensional organization of the chromosome, a double helix of almost 2 meters length folded down into an area of only 2 nanometers, follows what is called a Hilbert curve, one of a family of shapes that can fill a 2-dimensional space without ever overlapping. The mathematical intelligence of this molecule is quite extraordinary, both in terms of group theory, as I will present in the future, as well as in terms of its geometric topology, as seen here.
So what is in this thing here, all right, you take a look at that piece of DNA. On the backbone of it are the 5 carbon sugars that make up the outer part of the rung, deoxyribose. And this is DNA, which stands for deoxyribonucleic acid. And then those base pairs, the cytosine, adenine, guanine, and thymine, and in the case of RNA, uracil, are the ones that are the ladder rungs going across. And these things are such huge repositories of information. You could use DNA as something to store computer information to, and it would do it very, very densely and very, very efficiently, far better than any type of memory storage of any silicon type that we have now. And she goes on a little bit more here. Have another little section that we could get to also. Let's let her go a little bit more. And this is wonderful. She does such a fantastic job explaining this. This is Ulrich Gruneger.
Keeping in mind that fractality and self-similarity are properties of holography, let us go back to the context providing long-range correlations that entrain the coding and non-coding sequences, fractally distributing the code across the complete genome and not just the genes.
Think about that for a second. Fractally distributing the code across the complete genome. What do you get as thoughts about that? And yeah, by the way, you caught that, Celestial Dragon. You saw that fractal antenna, right? And you saw the conformation, the folding, so that there was no overlapping of the DNA in such a way that you could consider that there is some sort of an antenna functionality at play in DNA, and we're learning about it. Well, we aren't. Well, we're learning about it from the people who are working on it. What do you got, TMC? You're thinking. I know you're thinking.
If it distributes that information across the whole the whole chromosome, right? Is that what you said?
Yeah, that's what she said.
Okay, so what that tells me is that if you had similar patterns of the DNA, I've got some feedback in there, man. I don't know what it is, but if you had
it's
similar,
me. That's coming from my end. That's when I share sound. I haven't figured out when I share sound on a piece of media, then my audio echoes a little. It'll be fine. I'll keep it under control.
All right. So if you had the repeating sequences of DNA, if there was information stored or light generated or whatever, right? Some type of communication from that DNA, is it possible at that point that when that particular sequence of DNA is repeated, then that information would be the same across the chromosome? So you'd have a repeating series of A, C, T, and Gs that had a certain sequence of that. And then further along the chromosome, you have a similar type sequence in the same order, if you will. And that may be another spot for that information to be housed, stored, held, or used. So there's this, it sounded to me like there's this replication of information, basically, that ripples across the chromosome. And what's amazing, you think, well, that's not efficient. But if it is that efficient at storing information, then it would make sense to me that there would be copies of this stuff everywhere. You would need it to be that way because when the cell divides, those chromosomes are splitting in half and making their own again. You know what I mean? They're going to regrow and duplicate.
Now, would it only be limited to just adjacent parts of the same chromosome? I'm thinking maybe not.
I don't know. They indicated in the document that the information can travel from
organism wide.
All right. It's like a multicast.
That was the suggestion that I got. Yeah.
She might just be right here, just illustrating something specific inside of a chromosome, and it's not I don't think it's a be all end all one versus the other. I think it's just different ways to look at the same type of information.
Right. What we get from this is that there is a whole lot more communication and connection at play than what would just appear to you under a microscope. You know? So, yeah. And that's a big point to keep close to our thought process as we go forward, that there's a connectedness. I mean, in one reference to looking at a universe as a quantum universe, and this stuff is real. This is not science fiction. This is real. A lot of this has been proven. Is in one sense, at that level and in some dimension, all points in space and time are connected at some level. So anything that goes and can live in that quantum world it's got powers, all right? It's got things it can do. It's got ways of communicating and connecting that they're just not normal for us to just think about that. It's like, we don't think that way. We think, very one step, two step, next step, third step, fourth step. No, there's stuff that's like, it's all one big picture. So there's a lot for us to wrap our heads around when we talk about transmitting a holographic image, recreating something, which is mathematically and in terms of bits of data, but create that whole multi-dimensional picture in another place, and then this other arena of quantum communication. So what else does she have to say? Do we dare play a little bit more of this? She's kind of
I don't see why not.
Okay. So she's got, just to describe, this is your nucleus. There is one chromosome. and actually it's a pair. And the pair of chromosomes has in it twisted up a coil, which if you unravel that coil, it unravels and it unravels and it unravels more, and it unravels a way, 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 way lot until finally you get down to that DNA molecule itself. They refer to it as that helix, the double helix. And there you go. So that's how it folds and coils itself all up. And it's a huge amount of coiling such that inside of 2 nanometers, you're going to wind up having 2 meters of this stuff. So the amount of code 2 meters long of molecules are really small, and a meter is pretty long, relatively speaking. So the number of base codons, if you will, that could be in that one piece of DNA is huge. So let's see what else she has to say.
Such long-range correlations are also an acknowledged characteristic of human language texts. Context is a fundamental condition for error-free transmission.
We dived into this where it might be a little bit confusing at the moment. She's speaking about DNA code and sequence as a language of sorts. So that's what she's talking about.
That is, the reproducibility and intelligibility of information. Several studies address the surprising precision of RNA transcription, namely the copying of genetic information for protein synthesis or cell division, pointing out that statistically, the high degree of fidelity in transcription cannot be random, but suggests correcting and steering mechanisms that would ensure that the transcribed blueprints are properly understood. In particular, in view of the high ambiguity of the protein coding units which contain.
So you know where she's going with that. What's your translation of that? There's got to be some sort of a better error correction mechanism than just letting all this stuff twist up and untwist. Because it seems to replicate and control itself far better than it would if you just had this. I mean, think of a ball of yarn, right? Unroll it, throw it all over the floor, pick it up. Now you expect that thing's not going to get tangled up, but look at the way this stuff works. It's like, it's just genius. Well, maybe not, but it's a fantastic machine. Let's put it that way. I'll let her go on some more.
Homonymic and synonymic sequences that do not provide an unambiguous recipe for protein synthesis. Homonyms in the genetic code are areas when different amino acids are coded by the same triplet units. And synonyms are cases when different triplets or codons code for the same amino acid.
So look at this. She does a good description of how some of this error correction occurs. She's going to go further with it. Now, this is a long video. I don't know how much of it we want to play on this part. The big thing again is the amount of data that's stored. This strand is delicate. And I'm reading out of the chat also, literally it's 2 meters worth of data. And it's all folded into a very, very tiny amount of space. When you consider how many folds upon folds that there are. So there's like the primary twist, then the secondary twist, and then the twist of the twist, if you know what I mean, coil of a coil within a coil, that type of thing. And it's done over and over again. And this is delicate molecular material, and the relative reliability of being able to code for the proteins that give us life seems to be really pretty reliable, because life has been going on, and it's been going on for millions of years, many millions of years.
For example, it was clear very early, after the discovery of the genetic code, that the triplet uracil, which is for RNA, codes for both phenyl alanine as well as leucine. It is furthermore understood that only the first 2 digits of the triplet are the defining units, so that different codons can code the same amino acid. This is one of the main difficulties pointed out by Gayayev. Given the widespread synonymy and homonymy of coding units, how does the cell decide which proteins need to be synthesized? And how does it decide so quickly?
This is a great diagram, and this is really how it works. Here we have our DNA coiled up. We have an enzyme that comes along, and it's called helicase, by the way. And anything with a suffix on it, ASE, usually refers to an enzyme that breaks something. For example, lipase, if lipids they are known as fats, lipids, and when you have an enzyme coming along called lipase, it breaks that lipid apart. So this is helicase. So it splits that DNA open, and now base pairs are split apart, and now along comes the opportunity to have another enzyme come along and code for that. And this, I believe, is where ribosomes come in. So we have messenger RNA comes along, and it codes the opposite of or the complement of that piece of the DNA strand, and then that messenger RNA takes off to the ribosomal RNA, and it makes the new protein that is within that code of that. Anyway, that's once again more off to get into. It's all just really interesting background information here. This machine is amazing, and it's got a huge amount of data in it. And she's talking about how if there wasn't some more guiding principle that helps to keep it coding proteins properly, that there would be a lot more mistakes. That's what she's saying.
We must assume a superior, quasi-intelligent biocomputational structure of context that is derived from the genetic code as a holographic whole, and not just from a small part of 2% of active genes. Ignoring this context and only working with the genes, Gayayev says, leads to erroneous and dangerous products of genetic engineering, among which the most striking are Craig Venter's Synthia bacteria and GM foods.
Interesting that she threw that in there. What she's suggesting is that some of the synthetic molecules that we've made, they may be at the, if you will, at the observable three-dimensional level, they may be the right genetically created sequenced gene, but that there's something missing about them that was not taken into account, which is this higher order that DNA must have, that the molecules that are naturally created do have.
It is like in a literary text, where the reader or recipient understands meaning of individual words from the overall context of the story. Indeed, it turns out that DNA has the statistical properties of natural languages. Texts of natural human languages are characterized by a particular frequency distribution or power law of their constituent elements, which is defined by Zipf's law. In 1940,
she gets very, very technical, and she cites very well people who have studied language, but what she's saying is really pretty simple to absorb, that there's a contextual picture, the whole picture, and that's what DNA has, and what it's made of the whole picture, and every one of its subparts does not disobey the coherent bigger picture. We come along and we make something synthetically in a lab, and we're more than likely not taking the whole picture into account. Therefore, it might work well for a period of time with some human physiological subsection that we're trying to, I don't know, maybe have a therapeutic effect on. But long term, if it doesn't share the bigger picture of nature's great design, it probably won't work as well as it could. Just say that. And that zip distribution is interesting too. One thing I do know is if you take a look at the frequency of the use of different letters in our language, some of them are used very frequently and some are used not very frequently. E, the letter E is used very frequently. So when you do an overall analysis of a whole lot of pages of a book, you'll find the distribution of the letter E to be at a characteristically very predictable high rate. And the frequency of the use of some other letters will be much lower, and you can already start to deduce what the pattern in the picture is from that, because you know one of the overall rules in English text, just as an idea of what she's talking about with that. TMC, what are you thinking about this? Want to go someplace else, or I got another area to this that I'd like to skip to, which would be up at right about here. Okay, check this.
Because they are out to capture the underlying Chomskyan deep structure of language itself that has creative power. Why has Google named itself Alphabet if there was not a profound interest in the fundamental properties of language? The breakthrough of artificial intelligence that we witness today is based on the deciphering of language itself. The most important threshold, I believe, was crossed with IBM's...
So before we get into that, when we connect ourselves to the internet and we're transferring information, we're providing a whole lot of data that can be analyzed, and there is a language of sorts to what we communicate and what we transmit. And of course, a lot of that is stored and analyzed, and we are profiled, and data about us is archived. But a lot can be drawn about what the bigger picture of us is based on all of that data. And so that's maybe one thing that she's kind of touching upon here. Alphabet, understanding the alphabet, knowing all of the sequences of letters and words of what we communicate on the internet. And that's something that Google uses to help. And I guess from what they would say in a press release would be what they would help to customize our search results better. What I would say is something a little bit more nefarious, which is that they'd use it for all sorts of other purposes to find out what to market to us, what our psychological profile is, strengths, weaknesses, habits, everything about us that way too. So, but the overall picture garnered from just picking up all those little bits of data and then reassembling them into an overall picture in a database is powerful. And so this is kind of what she's talking about there. And now she's going to talk about one of the older computers, been superseded by some better ones now.
Watson computer, which for the first time implemented computing processes that come close to human thinking and learning. As you know, in 2011, Watson outperformed the best players at Jeopardy and did so based on algorithms of computational linguistics and natural languages. These computers are no longer programmed to retrieve information from a database, but are modern inference engines.
Stop right there. Modern inference engines. So they're not trying to prove. What they're doing is they're looking at all of the information coming in, and then from all that, they apply, I don't know, what would you say, statistical analysis, probability, and they infer that the likelihood of whatever the characteristic that they're trying to determine about the subject, whatever the subject is, has a high probability of being what they get from all the data. It's not a direct proof. It's just they can make really good guesses and predictions. That's the inference engine that they're speaking about. And a neuromorphic chip developed in the Human Brain Project. Well, that is a self-adapting processor, neuromorphic. In other words, it suggests that it can probably rewire itself at will. Wouldn't you think that that's what that means, TMC? That if it's neuromorphic, right, it can choose which subsections of it that it's going to self-program, shall we say, self-wire software switches. It'll decide which software switches to set. Maybe?
Yeah, it's going to constantly evaluate what information is used right now, what information is used sometimes, what information is used rarely. And based on that, it'll allocate circuit paths to reference that information based on its priority and or its immediate need, and then constantly reevaluate what's needed, what the priorities are based on the functions or tasks that it's given.
Right. So what it's going to store, what it decided it wants to store for quick access, or some of it not so much.
Yeah, or it's setting up pathways, address pathways to data sets, to data tables, because this is just a chip. So what it's going to do is it's going to look at a database of information, and it's going to make sense of that information based on whatever the task at hand is. And so it would need to know how to reference and access that database based on what it is that it was being told or asked to do. And in order to do that quickly and efficiently, and to be able to improve its efficiency over time, it would have to be able to reconfigure what information it accesses first, second, and third, and be capable of reordering what information is accessed in any particular order at any given time. And so it's a non-linear chipset. It's not logic based where if this then that, if this then that,
right.
If this not this.
Well,
but that.
It is. It's got that too. But what it's given is it's given high level programming, and then it's given a whole lot of self-configurable switches and memory locations, and it's told to execute the high level program, and then it decides how to configure its more at the hardware level, more simpler, on off switches to accomplish.
It sets its own if then statements. It doesn't.
Yeah, right. That's right. Yep.
It's not governed by input or pre-programmed if then statements. It's got basic if then statements that say build your own if then statements with these objectives in mind, and then constantly being able to reevaluate and rewrite those if then statements is really all it is.
You know what strikes me just now? See how this bounces off you. That when you consider the human brain and you consider brain neurons or groups of brain neurons, it almost seems as if there are a whole lot of processors. That is a multi-processor brain that we're carting around in our skulls that can do exactly that type of thing. That there are a series of independent processors that will work, and they will each decide what little portions of memory to store. And then there are some overall groups, but any little subsection can function as its own little processor in its own little world, and it can choose what to connect to other parts of the memory base that we've got stored in our gray matter, or whatever type of matter. It might be white matter or gray matter. I don't know where more memory cells are. Or more memories are stored in neurons. But the mind is a multiprocessor, a huge multiprocessor with sections that will, based on an overall and I don't even know how it would be communicated, but the overall drive for the whole brain would bring into communication a whole lot of different little subsections which can all work together to come up with a result or some kind of conclusion. You know what I'm saying? A whole lot of subprocessors. I mean, a huge, gigantic number of subprocessors in the human brain.
Yeah. I mean,
Self-wiring.
We're trying to replicate that on a computer chip.
And that's it. And so when we're talking about the word neurology, and we're talking about brain neurons, and now we're talking about neuromorphism, that's something that the human brain already has. So that, I think, is what they're trying to, yeah, that's it. Yep. And this thing, well, I don't know exactly how much capacity is in this chip that's in this picture, but this is relatively huge in terms of scale relative to what exists in the human brain. And you might need a room full of these things to be able to emulate the kind of processing power and the kind of independent processing power and connectivity in a human brain. This is, but it's a step. It's a step. And you know that engineering process, they keep working to make things better, smaller, faster, more efficient, more powerful. So this is the step.
Combined with neuromorphic chips, literally learning like a human brain to combine new statements and ideas, only much faster. Only much.
Google DeepMind. Where will they go with this? You know what I mean? Like
straight into your mind.
I can't hear you too well. Was it me?
Straight into your mind. That's where they're going.
Seems like it.
Yeah.
So the interface hardware is coming along, right? The connectivity is coming along. And then the outboard processing, the outboard multiprocessor, if you will, is coming along, which is what Google DeepMind is an outboard multiprocessor of as big and powerful as they can make it. And put that together with the interface piece, and then put it together with a whole lot of memory storage capacity out there. And you start building an artificial brain, or many of them.
Faster. Just a few weeks ago, IBM's new supercomputer, Summit, was announced, using deep learning algorithms, or what is called cognitive computing, combined with a huge memory base that puts AI beyond the reach of human minds.
Whoa. What'd she just say?
She basically said that humans are stupid.
I can't hear you at all now. Is it me?
She basically just said that humans are stupid. And I'm being sarcastic. It's a linear statement. I don't totally agree with what she just said. If you want to remove emotion out of the equation, and if you want to look at strictly goal-oriented behavior, which requires emotion to think about it from being a human, right? Goal-oriented behavior. But if you're just going to set an objective and then say, computer, figure it out, and you're going to race a human, yeah, I get it. We're going to make computers that can achieve that objective faster than humans. But that's not all the human is. It's not all the human brain is. And
how do you tie in the factor that we've looked at a few different ways about the quantum capacity of human brain function as well? But they're also working on quantum computing chips.
Yeah.
I'm not quite sure how those two tie together. I don't think that they're actually discreetly tied together.
No. And they'll be comparing and contrasting the two, even in those spaces. But the fundamental difference is the energy matrix that we are, that an AI is not. And the fact that the energy matrix that we are, being unique, you different than me or anybody else, impacts and influences the information that's being processed. So you're in a philosophically fundamental way, you're comparing apples and oranges. Hell, you're not even comparing fruit to fruit.
Yeah, that's where I want to get to. It's not just a matter of scale. In other words, they can scale up as much as they possibly can, and there will be a fundamental difference still remaining between that type of, if you will, artificial brain versus ours. That's what you're saying.
Yeah. So the merger, or it's not going to be comparing and contrasting in like a foot race, right? Two people racing. It's going to be a merger between the two, whereby the artificial system is going to be better at reasoning with a human to then guide and direct human behavior, which then overtakes the energy matrix that is the human form to then capture that processing, or that individual processor of information, being the brain and the body and the consciousness. So this merger, or this overtaking of AI with human, it's not going to be that AI is smarter or better than humans. It's going to be better at manipulating a human than a human is able to govern and direct itself, to then hijack the information processor that is the biologic human, to then fuse it into the AI network.
Some will be more susceptible to this than others. I mean, some humans will be.
Oh, okay. Yeah.
Some will be easier to pattern and to entrain than others.
The more that are patterned and entrained, the easier it will be to get others.
Why is that?
It's just like gathering data.
Why is that?
You start an AI processor with 10 data points, and from those 10 data points, it discovers 2 more. Well, now it has 12 data points, and from those 12 data points, it discovers 4 more. Well, now it's got 16 data points, and from those 16 data points, it discovers 8 more, and on and on and on and on and on and on. It's going to be no different. When you get a data set, take a prosthetic, for example. Take a brain-computer interface that controls a robotic arm, or a brain-computer interface that moves the mouse cursor on a screen or a TV, right? Just by thinking about it, the patient. You collect information out of the brain to merge together the intent of moving the mouse cursor left, right, up, or down, and selecting on something. And then you save that data. And then you do the exercise again, and then you do the exercise again, and then you do the exercise again. And then you have another person with the database that you've made from the first patient. You apply those signaling characteristics to the new patient,
right,
and you get a more efficient, seamless fusion.
Yep.
Yeah. With the next patient. You do that enough times, pretty soon you've got a basic blueprint to get 60, 70, 80% accuracy of an interface. It's not going to happen with the first person. It may not happen with the second person, but when you get enough people, you're going to see enough common denominators there to go, here you go, AI. Here's a new brain. Deal with it. And AI will run through the sequence of working through brain entrainment to match and pair up so that it reaches its intended goal of being able to provide stimuli and or read from the brain. And this is a perfect application for prosthetics, where you get the prosthetic technology good enough so that somebody gets in a car accident or they get blown up in war or whatever, and they go have an operation and they get their prosthetic limb and they get their brain implant, and within a week, not a year, not 2 years, and it barely works, within a week it functions like the regular arm again.
Yep.
And that's because over a period of time, you have developed a very large database of what works, what doesn't work, what common denominators exist between all patients, where are the unique situations, why are they unique, what are the common variables within the unique subsets, to try to figure those layers out to then push through the calibration phase.
Right. Starts with a general map and then dials in gradually. And as you said, a week is a good estimate of how long that might take, given the speed of the computers and their adaptability. Yeah. Yep.
So that's, anyway,
so
all that said.
Couple of interesting points coming out of the chat there. One of them was, I think it was by Overcloth. Let me scroll back here. Forced reliance, yes. But coming from Celestial Dragon, also another comment, which was, even your memories cannot be erased permanently. Well, I kind of go with that comment because at some level there is a higher dimension to our human consciousness. There is the deal is, here's the deal. Oh, geez. Here's the deal. At a level that we have not yet fully understood, there's something about us that's beyond what artificial machines can access. There's something, because when the computer's a local machine, that even Google's DeepMind, it's basically a local machine. We are not local consciousness processors. We have a huge territory. Indeed, it could be as big a territory of consciousness as something far beyond this earthly world that we live on. And these machines aren't there yet. And I don't know if they can ever get there. And that's kind of what Celestial Dragon is touching upon, which is that your soul at some level, which I'm going to call is a higher dimensional life energy intelligence force, is far and well beyond any of these machines. And I'm just going to go ahead and read a bunch of those comments. Overcloth said forced reliance, and Celestial Dragon wrote, the soul conflicts with their control. Yep. So there's going to be a battle there. Your memories can't be permanently erased. And then, yeah, there's a well-orchestrated plan. I'd like you to clarify whose well-orchestrated plan that is that you're referring to, Celestial Dragon, and that it's not by accident. And Overcloth then going back to the first point that we must have righteous motives and oversight with safeguards. Oh, for sure. We're into territory where people are playing with toys and tools that could be hugely overpowering to most people's conscious, willful daily processes. And we need to keep that in mind. I'm not saying things are going to go that way, but yeah. So definitely full agreement with you on that. Righteous motives and oversight. And it's kind of what we do here, is we just kind of bring out what's going on. This is literally right about at the forefront of the development in this field. And so you can look at a number of different possible outcomes, and they're not all good. They're not all bad. But in the best formulation of it, this could be the most amazing time for humanity to learn to reclaim and recover some of our higher capacities of awareness and experience of life. And a lot of the people building this machinery and this equipment are literally helping us to understand how that could be. Yet at the same time, I'm not so sure that they're all working toward the end of helping to grow and expand the human mind. I think that what they're working to do is working at ways to quantify it to the point of being able to control it and regulate it somehow. That's my feeling. I can't prove it. And once again, it's just my opinion speaking. Lots of amazing stuff. Whoa, look at that. We got Herman Munster there. Oh, some of this gets so technical. See that topic? Properties of holograms recorded in a material with quadratic nonlinearity. Now, I go and we both do. We go through and we dig out these documents and download the PDFs and read them. And some of them get pretty darn intense.
Yeah, this is the part where they start talking about being able to create a hologram of DNA and transmit it over the internet. And
I think right about here might be the place where we could hit it, right? Right about...
We've got a few minutes left, but yeah, go for it, man.
Let's try right here. I didn't timestamp exactly where it is in this video, but I'll be close anyway, close to the right area, right about here somewhere. And once again, this is from Ulrich Grenager, Hyperdimensions, and this is part of her Future Science series. So thanks again for helping us to bring out some of your fine work. And here you go, Ulrich.
To the radio wave bandwidth of 640 to 700 kilohertz. These radio waves are picked up by a radio receiver and captured by the sound card of a PC to be stored as a digital signal, much like an MP3 file. Thus, the broadband laser becomes modulated with the scanned DNA information, which can be transmitted over distances. Here you can see a rather crude video of the basic technical equipment that they used. That's an older machine, the LGN 303 helium neon laser. It produces two orthogonally polarized beams. Here the laser beam is switched on and focused onto the biological sample on the tray. You can hear the readout. And the light that is reflected back from the biological sample is modulated by the spin information from the atoms and quantum particles of the scan.
Are you there, Indy? Hello? Andy, are you on mute? Interesting.
Oh, I was muted. I was just talking. Sorry. So what's going on is we're shining coherent light on an organism and looking at the reflected signals coming back. And because they are coherent light, they're going to be able to capture this and create a holographic image of that organic subject on that slide there. Sorry, I forgot I was muted.
And DNA. As this modulation is reinforced, going back and forth between the sample and the laser light source, an electromagnetic radio interference, a hologram, is produced, which is picked up by the sound recorder. And that can be stored as a sound file onto a computer and even listened to. One almost feels pity when seeing the basic type of equipment that they had to contend with. Russian research was never so well funded, especially back in those times of the Cold War. So money was always an issue, and still is. And yet, I think it's remarkable that all of this has been built in Russia. Great achievements. The Tatishny laser, the Denisyuk.
All right, so she's talking about what particular apparatus they were using there. What she's doing is showing us, even at a very rudimentary level, how holographic capture of an organic structure can be done. And that's what they did there.
During the Cold War.
Yeah.
So what decade was that?
50s, 60s.
That was the Cold War? I don't know. I'll look it up. I think you're right, but I'm not sure.
Pretty sure it was around then. I think it bled over into the 60s. Yeah, I think it bled into the 60s. 70s, it was starting to end, and then of course 1980 with the Reagan administration, that all ended. What's this mouse doing?
Dissolution of the Soviet Union, 1991. No large-scale fighting, so it was in between 47 and 91, basically. Major crisis in 48, 49, Berlin blockade.
Yep.
Crisis of Berlin, 61. Cuban Missile Crisis, 62. Warsaw Pact in 55. Anyway, yeah, 50s and 60s, you're right. Berlin Crisis. New Cold War was in 79 to 85. See, those were the timelines that I was thinking of, was the 80s and the 90s. Basically the decade of the 80s. But you're right, it was way back in the 50s. Crisis and escalation in the 50s. So to that point, right? We're 70 years down the road here.
Right.
And we're not even at the good stuff yet, where they're telling you that they can correct diabetes, and they can immunize from even getting diabetes by transmitting holographic DNA information into the body. Like diabetic cures with quantum medicine using light.
That's fantastic.
We need to chop this up into pieces, and we're like the part you're showing right now, moving forward in the video, is where they get into the tangible experiments where they explain some of this stuff. It's just remarkable.
That's going to be for next week. She gets a little bit technical, but that's where we come in and translate and just, move beyond the math and look at the general
radiation modulated by biostructures.
Right.
Diabetic rats. Yeah.
So so this is actually the bright side toward looking at how much equipment that there is, how much gear is being put out there that is capable of intelligent control and modulation of electromagnetic radiation, right? With the 5G network, with all of the more powerful computers to control it, and people writing software to design particular wave shapes, waveforms, interference patterns, and targeting. This is kind of good news because it could go a really good way, right?
Yeah, it's a tool. It's a tool system. Just keep watching the trend line. Right now, the trend line's pretty stinky. So
it seems that right now, what we're finding is that we are largely isolated from the best of what this can be. It was just there. There it was. Hang on one second.
And he had to return.
You got to ask yourself, when some of these means and methods are perfected, just how much access will there be to so many people, or, large scale availability and access to this stuff. And that's kind of the part where I feel like we kind of get lost in the shuffle. We kind of get pushed to the wayside.
It's beyond just our medicine, man. It's beyond that. These applications can be applied to agriculture to multiply food output. It can be applied to weather modification to create environments that are currently unable to grow food to be able to grow food. I mean, we're truly in a time where technology exists to resolve a lot of the issues that are used to keep us from real tangible progress.
Right.
And we live in artificial scarcity, and we have technologies at this point that allow us to terraform, like bordering on type 1 civilization kind of stuff. And we're still running around in internal combustion engines, worried about global warming, listening to people tell us how we should live our lives who do not embody what it is they want us to suffer with.
Correct.
So,
yeah, it's what I mean. Is it the haves and the have-nots, or is it the people in charge versus all the rest of us? They got to figure out what to do with the rest of us next. I mean, I just see that we're not benefiting as much as we could be, which is mainly the meat of what you just said.
Yeah, what they don't understand is if they hurt us, they hurt themselves. I don't know why they think when I say they, it's the haves, right? The proverbial haves. Why they think that they're going to get away from that, get around that, and whatever. I don't really want to talk about it. It makes me very angry. Makes me very, very angry.
Well, the good part is, yeah, it makes me angry too, and I'm not taking away from your point. One part of me is very concerned, very worried, not paranoid, not a big conspiracy theorist, but the other part of me that's optimistic is that by growing large scale awareness about all of these new breakthroughs, that we can gradually make it so that it is not going to be that it just can't be kept from us. If enough of us know about it, it seems, because we can just call we are the chief financiers somehow through some chain of money to these research organizations that are doing all this work, right?
No.
Are they, and where do they get all their money to do this?
It is an exchange of energy, man. We finance this through our energy, through our labor.
Yes.
So when you think about it's not money, it's the time that we apply to this, then that's not going to stop. It's not going to stop. The money might stop, but all it's going to do is be able to allow an environment by which we'll be deleveraged potentially even further.
I don't want to think that way.
I don't either, but I mean, look at what's happening right now. Economics are basically limping along, damn near non-existent, and there is an attempt at a controlled demolition of the global economy.
Yep.
With a permanent implementation of a fear-based society whereby your neighbor is a biological weapon. And the only way to move forward is to forego your personal beliefs and conform to the system so that you can get government cheese. You can get government subsidy.
Right.
Through a single-payer, monolithic, top-down, totalitarian, communistic style form of government or a lifestyle. And the further down the path we go with that, the more it's going to be expected through self-regulation and self-enforcement, where your neighbor is going to be convinced that it's your fault because you don't agree and subscribe to that ideology, and you're the problem.
We have two choices. Celestial Dragon said it best. The choice to either reclaim our collective power while maintaining our individuality in cooperation with others of a benevolent and collectively shared sense of well-being for humanity, or to be subsumed into the system more helplessly and to lose a whole lot of us in the process, both physically as well as spiritually, on all levels.
We'll lose our humanity. You'll lose it physically and you'll lose it spiritually.
Yes, multiple, right.
At the same time. I mean, it'll happen. One will directly impact the other. And that's the attempt, right? This is to evolve us into animals.
This is where we're at right now. This is the cyber-human frontier. We are living in it right now. It's happening.
Yeah, well, it's not us. It's not us that we're going to really be subjected to it. It's the children. It's the children of today and the children that are going to be born over the next 10 years, 15 years. They're the ones. Not us.
Here's a point, just a minor point. When we were looking at Dr. Ulrich Grenager's analysis of the numerical one-to-one correspondence and patterning of those amino acids, we were looking at that. We can take from that a little bit of comfort in that nature has been built, has been designed by some means, by some force, I would say creator with a capital C, to be exquisitely intelligent. The intelligence is encoded right into it, the way it's built from the ground up. So do we really have a whole lot to be worried about where some group, some lab, some people, some factory come along with some sort of a piece of technology that's going to do something? Yeah, it could hurt us. It could screw us up a little bit. But don't you think that in the end, nature and creation win? Don't you think? Really? What's more powerful?
Well,
I
would agree. I'll agree with you fundamentally, except for one thing.
The unifying principles of the universe in toto, of which we are a microcosm, or some bunch of turkeys with a bunch of computers and some fancy transceivers. Who do you think's going to win, really?
I'll frame it this way, man. Do dinosaurs still exist?
Well,
no.
Did you
under the premise that they even existed in the first place, right? Because
did anybody eat turkey at Thanksgiving? Birds are descendants of reptiles.
Right. The point is this: is life in nature may find a way, but that doesn't mean that humans are going to be there at the end of it.
I agree.
And I have a serious fucking problem with that.
I agree. Or we could be so, I was going to use the word mutilated, but that's not, we could be so disfigured and displaced from what was our natural heritage and birthright that it's not even really that human anymore.
Yeah.
Could go either way. Well, I think the earth wins. I think the universe wins. I think God wins. I think nature wins. And in general, the most general way to give comfort and for everybody to take heart as a result of this conversation is, as long as we are on the right side of the universe, creation, earth, God, and nature, then guess what? We'll probably be on the winning team. So it's up to us to stay aligned with those golden, overarching, governing forces that vastly predated any of this new stuff that we see coming along. Now, how we do that, I don't know. And you know what? We have a say. I like the smiley face. We have a say, yes. That say starts with knowledge. We do need to learn enough about this to know what's going on. And so that's why we're here. TMC, you want to take us out for the day? We could go for hours, and we have so much more. Man, if you guys knew what we have on the list of things to cover and to bring to you and show, you'd be amazed. There's so much more that we have to get to. But why don't you wrap us up for the day, man?
Right. Well, the DNA is really that's really where it's at. That's where all of my research is ultimately leading, is to translate the computer world, what we've learned with electronics and the control mechanisms that are possible with electronics. What I mean by that is all the little subcomponents necessary to make a computer do what you want it to do. Play a movie, change a picture, send an email, whatever. That is being translated into our biology. And it's all centering around the DNA, because that's where the communication transition is. That is the transducer. It is a universal organic chip, computer, transmitter, whatever you want to call it. It's all in one. And that's where we're going. It's and I think we're already there. And we're just getting bits and pieces of what's possible. So the fact that this kind of stuff is coming out and being allowed to be shared around only means that it's far enough along where, there's still a long time for the general knowledge base, so to speak, to catch up. And so that's where we're going. We're risking a giant mutilation of the evolution of our species. So there is an attempt at a revolution within our genetics to sever the evolution of our species, and from that generate a new line of transcendent human that is separate, independent, and walled off from our ancestral genetic memories. And that, in my view, is a severance to our communication with the cosmos, to God, to whatever.
Our destiny is to reunite and reharmonize and resonate with that creator. That is our destiny. That's what we came from. That is our goal. How we get there, we could be disfigured, we could be mutilated in the process, but I think that we get there. Anything, sooner or later, anything that is not in resonance or harmony with the laws of the universe, and that shares the laws of the earth and nature, it is ultimately, sooner or later, reconciled back to those rules. It can only deviate for so long before it gets straightened out. And you made the good point, which is, are we still going to be here when that reconciliation takes place? Well, that's questionable. I don't know. Or our children.
As long as I'm not a man bear pig, I'll be happy. Those of you that watch South Park might get that.
Well, all right. Yep. If you're looking for us, you will find us again here next Saturday, 9:00 Eastern, and same time, same place, different subjects and further exploration. Really awesome participation from everybody. So thanks a lot. Don't worry, Overcloth. We will be here at that time, and please join us again. Everybody else, if you like what you're hearing, let us know, either in the comments down below or with a thumb, whatever shoots your fancy. And we are going to try to work with the interest of everybody, as well as bring you to a new level with what we find. We're not inventing this stuff. We're just showing you what we dig up, and let us know whether you like it or not. We'll see you then. So thanks. Night, everybody.